Hello everyone, welcome back to my CellChat video tutorials. Today I'm going to show you how to analyze cell-cell communication networks for sequencing-based spatial dataset. First, we load the package Surat, Tidyverse, and Patchwork. We use those packages to analyze the sequencing-based spatial dataset. So we are going to use the mouse brain sagittal anterior sequencing-based dataset. I already showed you how to analyze this dataset in my Surat video tutorial video 8 and 9. In Surat video 8 and 9, I didn't enable the cell clusters. So today, I'm going to quickly go through the data analysis and enable the cell clusters. If you want to know the details about how to analyze this dataset, you can watch video 8 and 9. So first, let's have a look at what's in the dataset. So if we open the dataset, you can see we have the array matrix data. It is a H5 file. You can download the data from 10x Genomics website. So the matrix data contain the gene names, they are the row names, and the cell names, they are the column names. And we also have a folder named the Spatial. If we open it, you can see in this folder there are 4 images and also a file named the scale factor, and an Excel to show the tissue positions. So we need to use the scale factor file and the tissue position file when we create the CellChat object. So we can use the load 10x spatial function from the Surat package to load the spatial sequencing data in. You can see we created a Surat object named the brain. We can plot the n count for the spatial sequencing data as plot 1. And also plot the n count on the images as plot 2. Then we can have a look at the wiring plot and the spatial plot for the n count spatial. Let's zoom in. You can see on the left hand side is the wiring plot for the n count. They are the total count for each spots. So on the right hand side is the n count for each spot. For the spatial sequencing data, normally a spot contains 5 to 6 cells. We can also use the wiring plot and the spatial feature plot to plot the n features for this dataset. Let's plot the wiring plot for the n features as plot 3, then spatial feature plot for the n features as plot 4. We can visualize them together. Let's zoom in again. You can see on the left hand side is the wiring plot for the n features, and on the right hand side is the spatial feature plot for the n features. So from the wiring plot and the spatial feature plot, we can see the n features and the n count. So we can go ahead to analyze this dataset. For the spatial sequencing dataset analysis, we can use the single cell transform function in the Surat package to normalize the data. After the data normalization, we run PCA. then find the neighbors, and find the clusters. After that, we can run UMAP. Now we are ready to look at the cell clusters. We can use the dim plot to see the UMAP for the cell clusters. We plot it as plot 5, and also we can use the spatial dim plot to see the cell clusters and the images. So we plot it as plot 6. Let's put plot 5 and plot 6 together and visualize it. We can zoom in. Now you can see we have 15 cell clusters on the left hand side, and also we can see the position of each cell cluster on the section. You can see each cell cluster locates on a distinct anatomical position. So we are going to subset the cortex area for cell-cell communication analysis. So you can see the cell clusters, cluster 2, 3, 7, 4, and 6. They are the cell clusters at the mouse brain cortex area. So let's subset the cell clusters from the cortex area to create a new threat object as the cortex. So you can see from our data windows we have a new threat object, cortex. So we can use the dim plot to check the cell clusters. You can see we only have the cell cluster 2, 3, 7, 4, and 6 on the UMAP. And also we can use the spatial dim plot to look at the cortex object. So let's zoom in to see the cell clusters in the cortex area. You can see we have the cluster 2, 3, 7, 4, and 6. So for the CellChat analysis, we need to label the cell clusters. We can have a look at the online tutorial to see how they label the cell clusters. You can see this is the threat online tutorial to analyze this dataset. We use the same method to analyze this dataset in our video tutorial. So let's scroll down. You can see the learning plot, spatial feature plot. And we scroll down further, you can see the cell clusters and the cell clusters on the image. So for the online tutorial, they use the reference dataset to annotate the cell clusters. So we are not going to use the reference dataset analysis today. To save time for today's demonstration, we can just name the cell clusters according to their annotation and also the cell clusters position in the cortex area. If we scroll down, you can see they labeled the cell cluster in the cortex region as astrocyte on the outside, L2, 3 neurons next to the astrocyte, then L4, L5 neurons, and also L6. And finally the oligodendrocyte. So to produce similar cell clusters as the online tutorial, we need to run the find cluster function again with resolution 0.9. We can have a look at the cell cluster now. You can see we have 9 cell clusters, and we can use the spatial dim plot to look at the cell clusters on the sections. So according to the position of those cell clusters on the cortex area, we can name the cell canisters, use the canister name from the online tutorial. Let's have a look at the name the cell canisters on the sections. We can zoom in. You can see we named the cell canister on the outside layer as the extra site. Then next to the extra site as L2, L3 neurons, then L4, L5, L6, and also the oligodendrocyte. So we named the cell canisters. Let's have a look at the surrogate object now. We can have a look at the cortex metadata. You can see we have the barcode in the row names. We have the uncount spatial and feature spatial. We did the analysis. Here are the cell canisters. And we named the cell canisters, but we don't have the canister name in the metadata. If we use the ident to look at the cortex object, you can see we named the cells. So we can create a new column in the metadata as the labels, then get the cell canister name information. We have a look at the metadata again. You can see now we have a column named as labels to have the cell types name for each cell in the cortex object. So now we can save the object, then we can use it for our cell-cell communication analysis. So let's clear our data window and also clear the plot. Now we can start to create a CellChat object. First, we need to load the packages for CellChat. We know the patchwork already, and we set the options. So now we can load back the cortex object. We can use spatial dim plot again to make sure we have a correct threat object. So you can see we named all the cell canisters. So now we can start to prepare the CellChat object. So first we need to use the get C data from the cortex object to create the data input file. So let's have a look at the cortex object. You can see in the assist note we have the spatial data. That's the original spatial data, and also we analyze the data. So we use the SC transform method to normalize the data. So we need to use the normalized data to create the data input. So let's create a data input file first. So next we can create the metadata. We can have a look at the metadata. You can see we have the barcode, the row names, and the labels column contains the cell type information. So for normal single cell RNA sequencing data, we can just use the data input and the metadata to create the CellChat object. But today we are going to analyze the spatial sequencing dataset. So we need to save the tissue image and also the spot position into the CellChat object. So we need to get the tissue coordinates from the cortex object as the spatial locus. And also I showed you we have a scale factors file in the dataset. So we can also load the scale factors information into R. You can see there are 4 data lists in the scale factor file. If we have a look at the file scale factor, you can see it contains 4 data. They are the information for the images. If we look at the spatial data set, you can see the scale factors contain the 9 image information, the tissue high resolution information, and the low resolution information. And also, we need to add the sports diameter as 65 into the scale factor. Here are the 4 data in the scale factor already, so let's add the spot diameter in. Now you can see the scale factors has 5 list data. So we have the data input, the metadata, and also the scale factor and the spatial locus for the sports position. So now we can create the CellChat object. So you can see we created a CellChat object for the spatial sequencing dataset. Let's have a look at the CellChat. You can see we have 17,668 genes, and we have 1,101 cells in the dataset. If we click the CellChat dataset, you can see at the moment we didn't add the CellChat database, and also the net and the netp data slot is 0. So first we can add the CellChat database into the CellChat object. Because this is a mouse dataset, so we need to use the mouse CellChat database, and we are going to use the secreted signaling database. So let's add the secreted signaling database into the CellChat object. Let's have a look at the CellChat now. You can see we added the secret signaling CellChat database into the CellChat object. You can see here only has 1,209 genes, but in our CellChat object we have 17,668 genes, so we need to subset the dataset to save our time for the analysis. After subset the data, we can see now we only have 562 genes to perform cell-cell communication network analysis. So now we can pre-process the data. First we identify the overexpressed genes. Then we identify the overexpressed interactions. After the pre-processing, we calculate the communication probabilities. You can see here, CellChat uses truncated mean method to analyze the spatial imaging dataset. After the calculation, so we can filter out the cell communication pathway if the signaling pathways is presented below 10 cells in each cell group. So now we can calculate the communication probabilities for the pathways, then aggregate the net pathways. So now we are ready to use the circle plot to look at the interaction numbers and also the interaction strengths. So let's plot the number of interactions and the interaction strengths side by side. We can zoom in to see the number of interactions and the interaction strengths. So here is the aggregated interaction numbers and also interaction strengths for all the signaling pathways in each cell type. And the width of the arrows show how strong the cell-cell communication network between two cell types. We can also see individual signaling pathways between different cell types. First, let's have a look at the CellChat object to see how many signaling pathways we identified in our analysis. So let's see the NetP pathway. You can see we identified 44 signaling pathways in this dataset. You can see here is the list for the 44 signaling pathways. We can look at all the signaling pathways. You can see we have the PTN, CCK, we have the MPY signaling pathway. MPY is a neuropeptide. Also, we have the SAM3 signaling pathway. It is axon guidance molecules. You can look at the signaling pathway that you are interested in. We can use the MPY and SAM3 as examples to show the individual signaling pathway using the circle plot. First, we can set the plot as 1 now, and we can show the MPY signaling pathway. We can zoom in. You can see now it's the MPY signaling pathways between different cell types. Next, we can use SAM3A signaling pathway as an example. Zoom in again. You can see this is SAM3 signaling pathway. There is a strong interaction between L6 and L5 neurons. So we can calculate the network centrality score. Then we can use the heat map to see the signaling pathways. So first, let's calculate the centrality score. Then we can use the SAM3 signaling pathway as an example again to see the cell communication using the heat map plot. Let's zoom in again. You can see L6-CT neurons express high-level SAM3A ligand, and the receiver cells, L5-PT neurons, express the high-level SAM3 receptors. Because this is a spatial sequencing dataset, the CellChat also has a spatial plot function to show the cell-cell communication on the sections. Again, we can use SAM3 signaling pathway as an example to plot the cell-cell communication network on the spatial image. So let's run the plot, and we can zoom in again. You can see now we have the cell clusters localized on the spatial image, and also we have the cell-cell communication network between different cell clusters for the SAM3 signaling pathway. I just used the SAM3 signaling pathway as an example. You can plot other signaling pathways. I think this plot is very beautiful. If you have spatial sequencing dataset analysis, you can analyze cell-cell communication network for your dataset and plot a figure like this for your presentation. This looks very cool. Okay, that's today's video tutorial. I'm going to stop here. If you are new to my channel, please subscribe to my channel and also share my videos with your friends. Thank you, and I hope to see you in my next video tutorial.